So hello everyone. So we have completed the process of DNA replication in prokaryotes up to initiation process. So now we are going to talk about the elongation of DNA replication. So we know that in this elongation process, the new strands of DNA are synthesized. So as I told you in the previous video that in elongation process, whatever events that take place in one replication fork, the same type of event take place in another replication fork. So for the convenience of explanation and just to make you more understandable, we have planned to discuss the elongation process in one of the replication fork. So whatever events take place in one of the replication fork, the same event take place in another replication fork. So we know that helicase enzyme is consistently unwinding double-stranded DNA, because the role of helicase is simply to unwind double-stranded DNA. So due to the unwinding nature of this helicase, both strands of DNA get unwound, and now when helicase unwinds the double-stranded DNA, it will create supercoiling beyond the region of unwinding. So this type of example we can simply practice at our home by unwinding the rope. So when you unwind the rope, it will create supercoiling at the top and at the bottom. So in DNA, same happens. So that supercoiling will cease the process of unwinding. So just to prevent the seizing effect of that supercoiling, there is another enzyme that is called topoisomerase, that is simply called DNA topoisomerase. So do you know the role of DNA topoisomerase? So DNA topoisomerase simply relaxes supercoiling. So whatever supercoiling is formed due to the unwinding of double-stranded DNA by helicase, so that supercoiling is relaxed by DNA topoisomerase. So we have to know that there are two types of topoisomerase used in DNA replication process: DNA topoisomerase type 1 and DNA topoisomerase type 2. So this DNA topoisomerase type 1 will relax supercoiling by cutting one of the strands of DNA, and this DNA topoisomerase 2 relaxes supercoiling by cutting the both strands of DNA. So this DNA topoisomerase 1 and DNA topoisomerase 2 has got nuclease activity as well as ligase activity. So this topoisomerase 1 means it cuts only one strand, and topoisomerase 2 means it cuts both strands of DNA and relaxes the supercoiling of DNA. So this is the role of topoisomerase. Now coming to our another major point, we know that the two strands of DNA are antiparallel. It means one of the parental strand must move from 3' to 5' direction, and another strand moves from 5' to 3' direction. So against this parental DNA strand, one new strand is formed, and against this lower parental strand, another new strand is formed. So at first we are going to talk about the formation of new DNA strand against this first parental strand, which moves from 3' direction to 5' direction. So For the process of elongation, there is an important enzyme that is called DNA polymerase III. So this DNA polymerase III has got some characters. So DNA polymerase III, when we look at the role of DNA polymerase III, this DNA polymerase III synthesizes new DNA strand only in 5' to 3' direction. This is the most important point that you have to remember. DNA polymerase III synthesizes new DNA strand only in 5' to 3' direction. One thing. Second point, DNA polymerase III cannot synthesize a new DNA strand directly. At first, there is a need of RNA primer. Once RNA primer is formed by an enzyme, DNA polymerase III, then can start new DNA strand formation. So that is another character is that needs RNA primer. Needs RNA primer for the synthesis of new DNA strand. And third most important point is that DNA polymerase III has got proofreading activity. This proofreading activity is called 3', 5' axonuclease activity. It is 3', 5' axonuclease activity. So this proofreading activity checks for the newly inserted deoxyribonucleotide at the new strand. So now we are going to talk the new DNA strand formation at this parental strand, which moves from 3' direction to 5' direction. So we know that the new strand which is formed against this parental strand will get polarity 5' to 3' because both strands of DNA are always anti-parallel. So this is our new DNA strand that is formed against this parental strand. But we know that DNA polymerase III needs the insertion of RNA primer. So this is the small fragment of RNA primer that is being synthesized by primase enzyme. There is the presence of primase enzyme that synthesizes RNA primer. So once RNA primer is formed by primase enzyme, DNA polymerase III, this is our, in this figure, this is our DNA polymerase III. So this DNA polymerase III will bind at the 3' end of RNA primer, and now it will synthesize new strand of DNA. So finally the polarity will be from 5' to 3' direction, which is possible by DNA polymerase III. So in this new DNA strand formation, we can see that the formation is quite continuous. That is, the formation of this new DNA strand is in the direction of the movement of helicase enzyme. So we say that when the formation of new DNA strand takes place continuously, that type of DNA strand is called a leading strand. So this is our leading strand because its formation is in continuous manner. So now, one drawback we can see here that at the tail of this new DNA strand, there is RNA primer. So as the name refers, RNA primer is made up of ribonucleotides, and in the structure of DNA, ribonucleotides should not be existed. So what happens that after the formation of this leading strand, this RNA primer is removed by one type of enzyme that is called DNA polymerase I. So DNA polymerase I, that is simply called Pol 1 we can say, or DNA polymerase I. So DNA polymerase I will remove this RNA primer. So after the removal of this RNA primer, there will be gap. So that gap is filled by DNA polymerase I only. DNA polymerase I removes this RNA primer and fills the gap. So after filling the gap, this the stretch at this gap and the preformed DNA strand are joined by the help of another enzyme that is called DNA ligase, and finally there will be formation of complete strand of new DNA, and this will finally generate one of the daughter DNA. So this is a leading strand. Now coming to the another strand, the major problem is at another strand which moves in which direction? 5' to 3' direction. We know that the parental strand which moves in 5' to 3' direction will get new strand in 3' to 5' direction. So the synthesis of new strand in 3' to 5' direction is not possible by DNA polymerase third. That's why DNA polymerase third will synthesize new DNA strand in opposite direction. So now we can simply understand this concept. So when we look at this segment of parental strand, if it is 3' end, so this will be 5', if this is 5' end, this will be 3' end, isn't it? So at this 3' end, RNA primer can easily be synthesized, and primase enzyme comes and put one RNA primer at this segment, and the RNA primer will get polarity 5' at this point. So once RNA primer is synthesized by a primase enzyme, now DNA polymerase will bind at the 3' end of RNA primer. Always remember, DNA polymerase third always binds at the 3' end of RNA primer. So once RNA primer is formed, at the 3' end of RNA primer, DNA polymerase third comes and bind and form new stretch of DNA. And finally, this will be the 3' end of new strand. So now, after the formation of new DNA strand at this segment of parental strand, helicase enzyme is continuously unwinding the DNA strands. So when further parental DNA strands are opened, again primase comes at this back side, and primase again put another RNA primer here, and after the putting RNA primer, this will be the 5' end of this RNA primer, and this will be the 3' end. So again, as we know, DNA polymerase third will bind at the 3' end of RNA primer and will form new DNA strand until it touches the first nucleotide of previous RNA primer. So as soon as DNA polymerase meets at the first nucleotide of RNA primer, the role of RNA primer stops. The role of DNA polymerase third will stop. So in this lower strand, we can see that new DNA strands are formed in small pieces, and the formation is also discontinuous. So the new DNA strand which is formed in discontinuous way is called lagging strand, and these small fragments of DNA are called Okazaki fragment. Now another problem that we can see here, that in this in between these small DNA fragments, RNA primers are present, isn't it? So one RNA primer is present between these two DNA strands. If helicase opens further more DNA, two DNA strands, so again this RNA primer will present between two DNA strands. So again there is role of DNA polymerase 1, that is DNA polymerase 1 will again remove this RNA primer. This gap is filled by DNA polymerase 1 only, and this newly added fragment is attached or ligated to the DNA strands by the help of DNA ligase enzyme. So these are the things that take place at the elongation phase, and this is simply called as the different events that take place at replication fork.